John here from bootcamp.com. Welcome to another video. In today's lesson, we're going to be taking a look at the spinocerebellar pathways, specifically the ventral and dorsal spinocerebellar pathways. We're also going to be taking a look at the cuniocerebellar pathway, which has a very similar function to these two other pathways. So if you want, you can pause and take a look at the outline, but let's just jump into it. So before we get into the details on how each pathway is structured, let's take a look at their functions and the levels or the dermatomes for where they're carrying sensory information from. So what does the spinocerebellar pathway do? Well, it carries unconscious proprioceptive information. So what type of information is this? This is basically how your brain knows where your limbs are in space with respect to your body. If I stick my hand behind my back, for example, while I'm sitting here, my brain knows that my feet are below me and planted on the ground and my arms are to the side. I don't really need to think about that type of information. And this information is coming from those stretch receptors, which are found deep in the muscles surrounding those muscle spindles. So that all that information is coming back and telling the brain what is going on with the muscles of those limbs without you having to think about it. So as you should be aware, the body is organized into different dermatomes. And you can see we have this diagram here showing the organization of the dermatomes. And the three pathways that we're going to be covering each carry a specific section of those dermatomes through that spinal tract. So the first pathway that we're going to talk about here is the cuniocerebellar tract. And we're going to talk about this one first because it carries information from the cervical area. And this is carrying unconscious proprioceptive information from the upper limbs, head, and neck, specifically from dermatomes C1 to C7. So if we look at our diagram here, that's going to be a lot of these dermatomes right here, C1 to C7. Now the dorsal spinocerebellar tract, this is carrying proprioceptive information from C8 to L3, specifically from the trunks and the lower limb. And if we take a look at the diagram, you can see it's a little bit here on the hand, that's C8, but the majority is coming from the trunk as well as the anterior aspect or the anterior upper aspect of the lower limb. Finally, we have our ventral spinocerebellar tract, and this is carrying unconscious proprioceptive information from L4 to CO1. And this is going to be specifically on the anterior lower limb as well as the posterior lower limb. So you can see on the anterior aspect of the lower limb as well as on the posterior lower limb itself. So you can see these different tracts are carrying information from different dermatome levels found throughout the body. And we can identify our dorsal and ventral spinocerebellar tracts right here. This is going to be our ventral spinocerebellar, and this will be our dorsal spinocerebellar. And you'll see that our cuniocerebellar tract is actually carrying information up the fasciculus cunatus in that dorsal column medial lemniscal system, but it's not necessarily part of it. Now, these pathways, unlike the spinothalamic and dorsal column medial lemniscal pathway, these only have two neurons. Now, starting off with our dorsal cerebellar pathway, it's going to start off similar to those other pathways in the sense that the cell body for the first neuron is going to be located in the dorsal root ganglion. From there, the axon is going to synapse on the second neuron in an area what is known as Clark's column, and this is a portion of the dorsal horn of the gray matter of the spinal cord. So Clark's column would be found somewhere right here, and then the axon from the second neuron, this is going to stay ipsilaterally, enter this dorsal spinocerebellar column, travel superiorly, and then once it reaches the medulla, it's actually going to enter into the cerebellum via the inferior cerebellar peduncle, which we have right here. So it's going to enter in through that inferior cerebellar peduncle, and then it's going to synapse in our cerebellum. So one thing you'll notice about this pathway is that it remains ipsilateral the first time. Both the first and the second neurons stay on the ipsilateral side for where sensation is being carried. So again, sensory information comes in, going to synapse in Clark's column. It's It's then going to remain ipsilaterally and travel up this dorsal cerebellar column or pathway, and then it's going to enter in through that inferior cerebellar peduncle and end up in the cerebellar cortex. And you can see on our diagram we have spinal segments. This one is going to be above C8, so this will likely be carrying information from the cuneo-cerebellar pathway, and this one is going to be below C8. So this is where we're going to have our dorsal and ventral spinocerebellar information coming in. So now let's take a look at the ventral spinocerebellar pathway, and this one is a little bit more detailed in the sense that there's a few decussations which occur throughout this pathway. So similar to the dorsocerebellar pathway, the dorsal root ganglion is where we're going to find the cell body for the first neuron in this pathway. It's then going to enter in and synapse again in Clark's column on a neuron in there. From here, the second neuron is going to decussate across and enter into that ventral spinocerebellar tract. You can see once it enters into the ventral spinocerebellar tract, it ascends up through the brainstem, past this area in the medulla, past the inferior cerebellar peduncle. So we're not entering the inferior cerebellar peduncle. We're going all the way up to the superior portion of the pons where it meets the midbrain, and we're entering into the cerebellum via the superior cerebellar peduncle. So now that we've entered into the superior cerebellar peduncle, now we're in the cerebellar cortex, but it doesn't end there. From here, it's actually going to cross back over to the other side, keeping that information ipsilateral. So now you can see we've crossed back over, and now we're ipsilateral again, and we terminate in that cerebellar cortex. So because this pathway decussates twice, it often confuses a lot of students. If we were to have a lesion in the spinal cord above the level from where it came in, let's say we have a lesion at around C8, we're going to see contralateral effects to that lesion, contralateral sensory deficits, because we decussate once and then we've lesioned the spinal cord after the decussation. So now the sensory deficits are going to be on the contralateral or the opposite side. If we were to have a lesion up here in the cerebellar cortex, then we would see ipsilateral sensory deficits because we've decussated, ascended, and then decussated again. So it's important to understand the intricacies of this pathway, and if there's a certain lesion somewhere, what sort of sensory deficits would you see? Finally, let's move on to the cuneo-cerebellar pathway. Now this one is a little straightforward. Begins similar to the other ones in the sense that the first neuron has its cell body located in the dorsal root ganglion. The axon is going to ascend in the fasciculus cuneatus all the way up to the medulla, where it's then going to synapse on the second neuron, found in the accessory cunate nucleus. So this structure right here and right here, these are going to be the accessory cunate nucleus, and you can see this is going to be found in close proximity to the nucleus cunatus as well as the nucleus gracilis. So once we've synapsed in that accessory cunate nucleus, then the second neuron enters in through that inferior cerebellar peduncle and terminates in the cerebellar cortex. So as you can see, the dorsal spinocerebellar and the cuneo-spinocerebellar pathways both remain ipsilateral the entire time. So if you were to have a lesion, you would see ipsilateral deficits to that lesion. Whereas the ventrospinocerebellar, it decussates, ascends, and then decussates again. So likely you would see contralateral deficits if you were to lesion anywhere along that track after the first decussation and before the second decussation. So now that we've gone over all these pathways, I've summarized them here just so you can get a sense of how they're organized and the different types of information that is traveling through them. So the dorsal spinocerebellar, remember that's this one in green. It comes in, synapses in Clark's column, ascends ipsilaterally in that dorsospinocerebellar pathway, enters through that inferior cerebellar peduncle, and then terminates in that cerebellar cortex. The ventrospinocerebellar tract carrying information from L4 to CO1. This one enters in, synapses on a neuron in Clark's column, decussates across, ascends in the ventrospinocerebellar tract contralaterally, all the way up to the level of the midbrain, where it enters into the superior cerebellar peduncle. Now that we're inside the cerebellar cortex, it's then going to decussate again and terminate in the cerebellar cortex. And then finally, we have our cuneo-cerebellar tract. This one comes in, ascends in that fasciculus cuneatus, synapses in the accessory cunate nucleus, and then enters into that inferior cerebellar peduncle and terminates in that cerebellar cortex. So again, when you're going through these pathways, walk through them in your mind to understand which ones are staying ipsilaterally and which ones are staying contralaterally, and what sort of deficits, either ipsilateral or contralateral, would you see if you had a lesion along the pathway. But anyways, that's it for today's lesson. I hope you all enjoyed that one. We walked through the dorsal and ventral spinocerebellar pathways, as well as covering the cuneo-cerebellar pathway. If you have any questions, always feel free to reach out to us, and I will see you in the next one.